Our first kind is called codominance or incomplete dominance. Remember before we said that traits are not inherited together, that they are completely separate, and that Mendel, in his time, people thought that if you had a red and a white flower, you'd end up with pink. Well, and we said that didn't happen. I kind of lied. Sometimes you can have alleles that are codominant or incompletely dominant, and this just means that neither allele is completely dominant over the other. Sometimes they do kind of blend or share. Places that you'll see this, you can see your pictures over there, would be flower type. Sometimes you do have, let's say you cross the red and the white flower, and you end up with heterozygous alleles. It might turn out as a blend of both, because like co-captains in a sport share that leadership role. Here, the alleles kind of share color. In mating cattle, there are some variations of cattle that you do have codominant or incompletely dominant alleles, and when they pair up, you end up with different colors. Multiple alleles is your pattern number 2. Multiple alleles means 3 or more alleles affective trait. So far we've looked at 2. A, capital, lowercase a. Capital T, lowercase t, etc. Some things like blood type are a little more complicated. Instead of big and little letters, they actually have 3 different options. Blood type uses the letter I, IA, IB, and I. You may or may not know anything about blood type, but there are 4 possible blood types: A, B, AB, and O. If your blood type is A, your genotype is either I capital A, or I capital A, I. If you're B, you're I capital B, or I capital B, I. If you are AB, your only option is IA, IB. And if you are O, you are little I all by itself. Polygenetic inheritance is your next pattern. And this is saying that certain traits, like skin color, and eye color, and hair color, aren't as easy as we make it. We were saying, okay, if you have blue eyes, you are little b, little b. If you are not blue eyes, then you have capital B, little b, or capital B, capital B. This is saying it's not that easy. Some things like skin color are affected by more than one gene at a time. So it's not as simple as one letter, skin color, let's say S. Okay, there's many, many different genes that work together to give you your skin color, and that's why we have such an array of possible skin colors in humanity, because there's more than just one gene that codes for it. A karyotype is a picture of somebody's chromosomes. What they do is they take a DNA sample, they isolate the chromosomes, stain them, and then look at them under the microscope. They take a picture, cut up the picture, and computers obviously do this nowadays, and rearrange the chromosomes by size from largest to smallest. Remember, you have 23 pairs, 1 copy each chromosome from mom and dad. And then we can look and see, oh, is there a problem here at number 18 or anywhere else with these chromosomes? Trisomy. Trisomy is when you have 3 chromosomes in a place where you should have 2. Trisomy 21 is Down syndrome. So if you looked at this karyotype and at spot 21, somebody with Down syndrome would physically have a 3rd chromosome in that spot. This is a nice little animation that shows what happens during trisomy. So we have all of our chromosomes. Here's our egg cell, and somehow we ended up with an extra copy down here. So now we have 24 instead of 23 in this egg cell. Okay, we have a sperm that's coming. The sperm has the normal amount of chromosomes, 23, and they're going to show us that next. Whereas the egg accidentally had 24. The sperm fertilizes the egg, so the 2 cells fuse together. And now we have our chromosomes, except we have this one that has 3. That is trisomy, and then depending on where the 3rd one is located, we assign a number to it. Trisomy 21 would be 3 chromosomes at spot 21. Monosomy would be if you are supposed to have 2 chromosomes in a place, like you are supposed to, there's actually only 1 there instead. Same animation, except now we're going to see monosomy. Egg cell created during abnormal meiosis is missing a chromosome. We only have 22 instead of 23. Here comes the sperm, and the mutation could easily be in the sperm as well, not always the egg here. The sperm comes and fertilizes and has 23 chromosomes, the normal number. They fertilize each other, meaning they join together. And you can see here in the baby's first cell, we're missing one. That is monosomy. We already learned about DNA fingerprinting with the lollipop NOVA exercise. DNA fingerprinting is when we take DNA, we cut up the pieces, we run them through a gel, and then the electricity sorts the pieces by size. We can use this for paternity testing, solving crimes. So like in this picture here, the child is going to have a mix of DNA from mom and dad. It got this gene from mom. This gene came from Larry, as did so this Larry's obviously the father. Bob has a gene here which did not get passed on, so that would mean he's not the dad. Here's a real picture of a DNA fingerprint. Pedigrees, or family trees, are used to track a trait through generations. Squares are boys, circles are girls. If somebody is filled in, they have the disease or the disorder, and somebody who is empty does not. Here we have 4 generations, so these would be the parents, and this line connecting them means they're married. These drop-down lines are their children. So this 2, 3, 4, 6, and 8 are their children. And then these people that are connected by a horizontal line married in. Take a look here. This one's a little bit different. It shows half-shaded boxes. That would be somebody who is heterozygous or a carrier. So instead of being big A, if that's the letter we're using, this would be big A, little a. They're a carrier. What do you see going on here with this double line? If you look down here in the key, it tells you that they're married and related, so this would be incest. This pedigree has some fun symbols on it, okay? So incest. This here, this triangle, is telling you that there are twins, and you'll learn some more of these symbols as we go through the pedigree project. Genetic counselors. These people interact with patients and communities, and they do basically research on a family's genetic history and take into account how likely are certain traits are to get passed on, and then counsel families if they should have children and whatnot. If you're interested in this career, it's actually one that I was considering for a while. It's a 4-year bachelor's degree, normal college term, and then 2 years after that of graduate school, which is a master's degree. Some neat places nearby that have this would be Madison and the University of Minnesota. Salary, if you're interested in salary, medium income is in the $60,000 range. On the high end, you can make over $70,000. A cytogeneticist is somebody who uses microscopes and would do things like karyotypes and analyzing chromosome samples and things by looking under microscopes. This is a 4-year degree, followed by a 1-year program, which can also be done as you're completing your 4 years. Different places nearby that do this: Mayo Clinic in Rochester, Madison, Marshfield, Milwaukee. Medium income, again, around $60,000. On the high end, almost $70,000. Your next step is going to be looking at pedigrees, and your project for this unit is creating a pedigree for a family. After we're done with that, we're going to look at how genetics and heredity relate to us in other ways. Please look at your note packet for what your next steps are, and as always, if you have any questions or need any help along the way, please ask.